Hello, everyone. My name is Shahab Bakhtiari, and I'm an assistant professor at University of Montreal. And in the NeuroAI OHBM workshop in Montreal, I'm going to talk about functional specialization in biological and artificial neural networks. And specifically, I will be talking about how we use the deep learning approach and artificial neural networks to model the visual system. And specifically, I will focus on the fact that once we use artificial neural networks to model the visual system, we need to be able to learn the same sort of a specialization that exists in the brain in the neural network. So I'm going to talk about the type of a specialization that exists in the visual system. I will briefly describe those. Also, I'm going to talk about the methodology that we and others have been using to compare neural networks and artificial neural networks and the visual system. And specifically, I will talk about the recent steps toward mapping different specializations between the visual system and the neural network. So, we are, this is a NeuroAI workshop, and we are basically doing research in this space, which is at the intersection of deep learning and neuroscience. The research in this field goes into different directions. We use artificial neural networks and deep learning tools to model the brain, to develop models of different systems in the brain. And also we take inspirations from the visual system specifically and the brain in general to develop better deep learning, better AI, better neural networks. And in the work that I'm going to talk about today and the literature that I'm going to summarize in this talk, we're using artificial neural networks to model the visual system specifically. So, well, the visual system is one of the most studied and investigated parts of the visual system, the brain, and we can say that probably it is also one of the best understood part of the visual system. We know a lot about the organization, the anatomy of the visual system, the functional organization of the visual system. The tuning properties, receptive fields, and other properties of this system also in different species, in humans, other primates, in rodents, cats, other carnivores, and so on. And so specifically, we know that in the visual system we have two parallel pathways. So the ventral or the what pathway is in charge of object categorization, scene understanding, face recognition, basically dealing with the static content of the visual experience. And this is always, at least in the in primates' brain, in this case, the human brain, it is an occipitotemporal. Anatomically, it's an occipitotemporal projection, as you're seeing here. There's also the dorsal or the ver pathway, which is an occipital parietal projection. And this part of the visual system is mostly in charge of localizing objects, tracking moving objects, motion perception, guiding actions and movements based on the visual input. And these two pathways have different specialized roles, and the differences between them and their functional properties are very well studied in the last decades. So now we are working in deep learning framework in the NeuroAI field of research. We are functioning within a deep learning framework. So the hypotheses that we build are within this deep learning framework. So in this framework, we're always working with neural networks. Neural networks have architectures. There are different types of architectures of neural networks. We have the convolutional neural networks. We have the multi-layer perceptrons and other types of architectures. Most recently, we have the transformers that have shown great improvements in the performance of these models. So we have this architecture of the neural network. We also have the objective function, which is the loss function, the reward function, or other type of objective function that is optimized in order to train the network, tune all the parameters of this network to do the specific task or show the specific behavior that we want it to show. We have also the learning rule, which is basically telling us how exactly the parameters of the network should change, that all the connections between different artificial units in the network should change depending on how the objective function is changing. So in the majority of work in deep learning and the fields that work with deep neural networks, in the majority of works we are using backpropagation algorithm, but there are also other learning rules, some that are more biologically plausible, that one could potentially use to train these neural networks. Their function is not the performance of those other learning rules is not as efficient as backpropagation. So backpropagation, because of its success, has been basically dominating the whole field. And also we have the data, right? The data that is being used to train the neural network for the specific objective function and specific behavior that the neural network is trained on. So when we put all these, all these components are, they basically set the decisions that we have to make during designing a neural network. We have to decide about the architecture of the neural network, objective function, and so on. Then we make all these decisions, put them together, we make a system that at least sometimes it seems to be able to show some intelligent behavior. So once we're functioning in this space, of deep learning, to model the brain, then we want to choose all these components in a way that give us the best model of the brain. In our case, in this talk specifically, give us the best model of the visual system. I'm going to basically use this framework to talk about the advances, the recent advances in modeling a specialization of the visual system. So before going into the next steps, I'll talk a bit about the methodology. This approach or this research question has been asked in different species. The neural data have been collected from different species, the visual system of mice, using data modalities like two-photon calcium imaging, electrophysiology, and the same goes for monkeys, mostly electrophysiology. Very, very recent work that I saw in doing calcium imaging in monkeys, but in 99% of the work, we're talking either about electrophysiology in monkeys or fMRI. And in humans, it has been mostly fMRI with some MEG and EEG work. So the data for building these models is coming from different species, different data modalities. So we want to basically take this data from different species, and we want to also build neural network models of the visual system of these species, and we want to compare to see how good this model is in terms of similarity with the visual system in these different species. And in general, we have from the brain of all these species, basically regardless of the type of data that is collected, always the type of data that we're working with is recorded brain activity in response to some stimuli. So I'm showing this with Y here. These are most of the time time series recorded either from single neurons or from regions of interest in different species, and in response to different types of stimuli, different numbers of stimuli, visual stimuli. And on the other hand, we have the activations collected from all the artificial neurons in the neural network in response to the same type of stimuli. So we have these activations of the neural network. We have the data recorded from the brains of these species, and we want to basically develop a neural network model and compare the similarity between them. But in order to compare the similarity between a developed neural network and the visual system, there has been different methods, different measures of or metrics of similarity. There are the regression-based metrics that are simply fitting a linear model between the activations of the neuron, the artificial units in the neural network, and the data recorded from the brain of different species. So it's basically fitting a linear regression model that uses the activations of these artificial neurons in response to the stimuli for predicting the neural activity recorded from the visual system of an animal. And then when the linear regressions fit, this measure of correlation is used to tell us basically how good this model is in predicting the responses of a certain brain area, for example. This is the correlation between the predicted and the actual brain data given this model. This is a regression technique, and it has been used in several studies and still being used. I'll talk about some of the cases that this method has been used in. And I'm not going into the details. There are upsides and downsides in all these metrics that another talk in this workshop is going to cover it. The talk given by Dr. Elizabeth Dupre is going to cover all these similarity measures and their basically drawbacks and upsides, downsides. I'm only discussing very briefly the basics of these similarity measures. So another similarity measure that is very commonly used in this field is representational similarity analysis. So in this measure, we basically have the responses of neurons in brain area. We have the responses of neurons in response to different visual stimuli. So in this high dimensional space, in a cartoonish way, I'm only showing 3 dimensions, but if every dimension you imagine that this is a neuron, in this very high dimensional space, you have a single point per image that is shown to the subject. And from this data, we basically have a matrix, which is a response matrix, response of all the recorded neurons to all the stimuli that we have, via a measure like YY transform. It's a sort of inner product between it's an inner product of responses of all the neurons to all the stimuli. We get the representational similarity matrix. And in this matrix, every element is telling us how similarly every 2 stimuli are represented in this specific brain area. Okay, so this matrix is kind of summarizing the geometry of visual representation in brain area. You can do the same thing for a layer of your neural network, right? You can collect the responses of all the units in a specific layer of your neural network to the same stimuli and build the representational similarity matrix for that certain area. And then what you can do is that you can take the representational similarity from the brain and from the neural network and calculate some measure of correlation between them to see how similarly this brain area and this layer of a neural network are representing this sort of stimuli. And this is the general idea behind representational similarity analysis. There are different variants of this method in terms of the distance metric that they use, the similarity measures, and they're all I would say that the results are not agnostic to these choices. So Elizabeth is going to describe all these important points that we have to consider when we're doing this analysis. But this is basically the gist of this method. So we use this type of method, either the regression-based methods or representational similarity analysis to compare the neural networks and the brain. So we are interested in building, as I said, building a neural network model of parallel pathways of the visual system. So we have the data that I said it's coming from, it can come from monkeys, humans, or rodents. We have the measure of comparing a neural network with brain data, and we want to build a neural network that is giving us the highest similarity. And as I said, we are functioning in this deep learning framework. And specifically in this talk, I'm going to mostly focus on the objective function. and how choosing the objective function can give us a model of different specialization in individuals, and how it is important in developing models that are matching with the specializations of the visual system. I will also touch a bit in the last part of time. I'm going to touch a bit on the architecture and how that might play a role. And I'm hoping that this will give you a general overview of how we think in this framework and how we test different hypotheses in this framework. So, so the first thing is that we want to build a model of the ventral pathway. And this is the work that that was done a few years ago. And the hypothesis was basically, and the hypothesis was driven by a lot of work that came before that. But at some point, after the revival of deep learning and neural networks, that hypothesis was kind of translated to this format, this kind of deep learning framework. The hypothesis is that ANNs that are trained with an object categorization loss develop what representations similar to the ventral pathway in the brain. And so to just have an idea of where this hypothesis is coming from, for many, many years, people had shown and had investigated that the ventral pathway in primate visual systems specifically is in charge of object representation and object discrimination. So that idea was translated to this hypothesis that now if we take a neural network and train it with object categorization, it should give us a good model of the ventral pathway, given that the ventral pathway is also in charge of object categorization. And that was basically this hypothesis was at the center of this work by Danny Ammons and colleagues in 2014. And they basically took a neural network, and the neural network was optimized to do object categorization. And then they compared that neural network with recordings from different areas along the ventral pathway in monkey visual cortex. So the way they did it was that they had the recordings from these areas of monkey visual cortex. Specifically here, they're showing the responses in area IT. This is a big part of the ventral pathway, is one of the last stages of processing in the ventral pathway. So they had just recordings from area IT in response to different images of objects. You know, you're seeing some samples here. So the black traces underneath these red traces are the responses of the neurons, and the red traces are the responses of neurons of a deep neural network in response to the same stimuli. So the match, if you just look at this figure here, the match is showing you a very good predictive capability of neural networks in explaining the responses of neurons in area IT. They also showed that so if you just look at these red bars here, red histograms, you're seeing that as you go deeper in neural network, so the first layer, second layer, and so on, the top layer is giving us the highest explained variance of neural responses. So the toppest layer of the network is the best layer that can explain the responses of area IT. And given that area IT is kind of the last stage of visual processing in the ventral pathway, this was a very great demonstration. They also compared that model with previously developed models, the models that were developed before that. So, you know, we're talking about 2014, right? So these are all the models that were developed before that, previous decades, previous years. And they could basically show that this shown in red here, which is the neural network model, they call HMO, stands for another thing, but just imagine that this is the neural network model. And they show that it can outperform, specifically the top layer can outperform all the previous models. And this was a large margin of overperformance compared to the previous developed models. The other important demonstration, which is great evidence for the hypothesis that was postulated, was that they showed a very, very strong correlation between categorization performance and IT explained variance. So basically what they're showing here is that the better the model does on object categorization, the better it does in predicting IT responses. This very, very strong uh, correlation. And this is the support for the hypothesis that if you have a model and you train it on object categorization, it turns out to be a good model of at least area IT in the ventral pathway. They also did the same kind of analysis for V4. And as you can see here, it's showing you something interesting. It's showing that for the red histograms here, you can seeing that the layer 1 is not doing very well. The intermediate layers are doing relatively good, and the last layer, the top layer, is not doing as well. And this is it's a very nice demonstration showing that the intermediate layers of the neural network are the best predictors of area V4. And that makes a lot of sense given that area V4 is an intermediate stage of visual processing in the ventral pathway. And this is a nice proof of concept. And since that work, benchmarks like BrainScore have been developed where you can submit your neural network model, and the benchmark will basically rank your model based on the performance of the model on predicting the responses of different areas along the ventral pathway. And mostly these models have been different in terms of the size of the models. More recent models are way larger, and also the architecture of these models are different. The first models were like AlexNet that had a relatively simple architecture, but as models were getting better and better, there were more nuances in the architectures. And you can see that, for example, you can go and check on BrainScore, see which models are now the best models in terms of modeling the ventral pathway. But it's also important to pay attention to this recent work from Lindsley et al., showing that now if you look at the recent developments in deep neural networks, the pattern that we observed before, showing that the better the model does on object categorization, it does better on predicting neural responses and along the ventral pathway. For the recent developments, it doesn't hold anymore. So we can see that the performance on predicting the ventral pathway response is actually getting a bit worse. Although the performance on ImageNet, the image classification or object classification data set, is getting better and better. It's passing 85%, getting close to 90%. Neural predictivity is actually going down, which suggests that potentially object categorization as a loss function is not enough for developing a good model of the ventral pathway. So we can't just push on object categorization performance and expect to get a better model of the ventral pathway. This is a very important observation, at least in the way that we define object categorization by the ImageNet dataset. This seems to tell us that there are more complexities and nuances in the story that kind of opens the path for the future research. But in all this work that had been done from Jim DiCarlo's group at MIT, Dan Yamin's group at Stanford, and others, and also the fMRI human research fMRI results from Nico Kruskordi group and others, they kind of all pointed in the same direction, that models developed for object categorization can be a good model of the ventral pathway. And in all these works, in almost all of them, the visual system was kind of assumed to be this part of the visual system with this very one pathway, strictly feed-forward architecture. But in reality, the visual system looks more like this diagram from this famous diagram by Fleman and Van Essen in the early 90s. So from this figure, we can see that the visual system has a modular organization, it has parallel pathways, and it has functional specialization. And this is something that you can't see it from this figure, but this is something that has been shown before, that there are specializations. As I said, at least we have the ventral and the dorsal pathway in the visual system. So again, we are dealing with, they're basically working in this deep learning framework. And the question that we want to answer now is that how can I have a model, a neural network model, that also has this sort of parallel architectures and parallel especially pathways, as we have in the visual system. So to develop a model of a neural network model of the visual system, we could take two approaches. So I'll describe both, and I start with this approach. We can assume that we have two pathways, right? So we could assume that we have two neural networks representing each pathway of the visual system, and each of them has its own objective function or loss function, right? So for the ventral, the network that represents the ventral pathway, we already showed that object categorization can do a good job, a relatively good job in modeling the ventral pathway, at least from what we have so far. Now, the question is that if it's assumed that the other pathway, the dorsal pathway, is a neural network with another loss function, what would be that other loss function? So in a recent work, we hypothesized that you can basically train a neural network with a self-motion estimation loss. And that neural network will develop wear representations similar to the dorsal pathway of the brain. And again, similar to the previous hypothesis about the ventral pathway and object categorization, this hypothesis also had a history from the studies of the dorsal pathway in primates. People had shown that the responses along the dorsal pathway in primate visual system is very important for self-motion estimation and self-motion guidance, right? So as you're moving around in the world, you're walking, you're driving a car, from the visual input that you're receiving, you're able to estimate the velocity and direction of your own motion. And this information about self-motion, people had shown that it is represented in the dorsal pathway. So that history of studies was kind of translated to this hypothesis in a deep learning framework. Now we said, okay, let's train a neural network on self-motion estimation. And the hypothesis is that it should develop a dorsal pathway-like representation. So to do that, we took a 3D convolutional neural network, and as input, we gave the model a sequence of images, basically a short video, and the videos were simulated videos. For that simulation, we basically put a camera somewhere in the world, in a simulated world, and we moved it with a specific velocity, in a specific direction, and gave that short segment of video as input to the neural network. And at the output, the network should be able to estimate the parameters of camera motion or self-motion, right? So we did that, and then we took that model and ran a bunch of comparisons. So as I said before, most of the comparisons between neural networks and the brain, different areas of the visual system, were based on either regression-based metrics or representational similarity analysis. But there's also another approach, which is a bit indirect. It is not as direct as fitting responses of the neurons and artificial units, but rather it is actually about comparing the functional properties of the neurons in a brain area versus the neural network, the artificial neural network. So this is what I'm showing here, and we did in the first part of the analysis in this work. So here, for example, I'm showing the tuning curve properties of the neurons in the first layer of our neural network. I'm not going to go into the details of this, but basically what we show here, and it needs a bit of familiarity with the literature of motion tuning, for example, in the dorsal pathway, but this exactly shows the type of tunings that you would expect from V1 neurons in response to moving stimuli. So layer 2, which is very similar to the dorsal pathway, also shows tuning properties. So these are histograms that show the tuning preference of average neurons in layer 2, for example. And here again, these tunings are similar to what we expect to see in area MT of the dorsal pathway. And layer 3 is showing us the type of tuning properties that are expected to see in area MST, kind of the latest stage of visual processing in the dorsal pathway. And also, when you look at the spatial filters of the network after training, you see the type of motion tunings that you would expect to see in the dorsal pathway along different stages of processing. But also, we did the classic type of linear regression fit between the responses of neurons in V1 and different layers of the network. And here we are benchmarking, we are comparing across different models. So every row that you're seeing here is just one layer of the model that we have. And as you can see here, in almost all cases, our model, except for V1, our model, the dorsal net, what we call the dorsal net, is outperforming the other models, shown in bold font here. And this is kind of showing that with this specific measure of similarity, also our model is outperforming the other models, comparing the responses of these three areas along the dorsal pathway. The other important feature is that when you compare across layers of the neural network with different areas, we can see that the highest areas of the network are better for explaining area MST, the mid layers are better for area MT, and the first layers is better for area V1, showing the same kind of hierarchy that we observe in the brain, in the visual system. In another work done by Rido and Welshman, they kind of showed the same principle, but in another way, with another approach. Here, again, they have a neural network. They call the neural network motion net, which is the multi-layer neural network. Now, as input, they have these static images that they move them in a specific direction, a specific velocity, and with this movement, they create a very short video segment. They fit that to the network and estimate the motion direction of the movement in that video. So here it's not really self motion. It can be considered as object motion, but still the network is trained to predict motion. And here again, they go and compare the tuning properties of the trained network with what we expect to see from areas, specifically area V1 and MT as part of the dorsal pathway in primates. So what they're showing here is the neurophysiological data from the neurophysiology, and this is the tuning properties. So this is the histogram of the preferred motion direction across neurons in V1. And this is something that they took from some neurophysiological data, recordings from probably from monkeys. And here is what they can get from their motion net. So what is very important in this figure here from the neurophysiological data is that the majority of neurons are preferring oblique directions. They're mostly responsive to horizontal and vertical motions, to cardinal motions, and less responsive to oblique motions. And you can see the same thing in their model. Again, most of the neurons are responding to horizontal and vertical motion. And the idea is that this sort of preference in the fact that the majority of the neurons are responding to cardinal motions is coming from the statistics of natural images. So they rotated the natural image by 45 degrees, and now they show that the majority of the neurons are responsive to oblique directions. So basically, the preferred directions of motion also was rotated 45 degrees. So their model is also able to capture at least this part of the property of responses in neurons. They also compared an important property of responses of neurons in area V1 and area MT. So importantly, when you have two stimuli that are called drifting gratings, some sort of Gabor patches that move in a specific direction. If you overlay two of them together, moving in 90 degrees difference, what is perceived usually is an angle that is between these two angles. And it has been shown that V1 neurons mostly respond to these so-called component motions, while MT neurons, a higher area of the dorsal pathway, responds to the pattern motion. So this is what is shown here. MT neurons mostly respond to the pattern motion, responding to the angle between the two angles, the two component angles. And the V1 neurons mostly respond to the component angles, shown in pink here. And they show the same thing for the V1 part of their model and the MT part of their model. So this is, again, another electrophysiological property of the neurons that is replicated in their model. So, together, these works are basically telling us that you can have this motion estimation or self-motion estimation as a loss function or objective function, and you can train a network with that objective function, which then gives you a model of the dorsal pathway. So with these works, you can have neural networks, separate neural networks that are trained with different objective functions, but they can capture the specialization in the visual cortex. Now, what if we want to develop the same sort of two-pathway model, right? But can we have one single loss function? Is there any specific loss function that once a two-pathway network is trained on, it can automatically develop the two types of specialization, the dorsal-like and the ventral-like representation? So this is a question that we tried to address, and we came up with this intuition that visual prediction, in order to predict what happens next in our visual experience, requires two different and competing forms of representations. So imagine that you're looking and in front of you, there's a car moving in your visual field, right? And our visual system, as shown in previous work, is quite good in predicting what happens next in the visual experience, in the visual field. So now the car is moving a specific direction, a specific velocity, and our visual system wants to predict the motion of the car and where the car will be in the next moment, right? So there are two types of prediction that the visual system should be able to handle. One is what we call movement invariant prediction. So it basically is that I will see an orange car, right? No matter how it moves, I will see a car in the next moment because it's in my visual field, it's moving, but the car is car. So it is invariant to motion. This kind of, recognizing and predicting the identity of the object is something that the ventral pathway is good at. But also, the visual system should be able to predict the motion of the car, right? So no matter what the object is, this visual system should be able to say that there will be a leftward movement. And this is, again, something that we know the dorsal pathway is good at. So with this intuition, we hypothesize that if you just take a neural network and train it on visual prediction with a self-supervised learning approach, it should spontaneously develop the two types of representations, the ventral-like and the dorsal-type representations. So we tested this hypothesis. We developed this this kind of training framework in which we are using a method called contrastive predictive coding. It's a self-supervised learning approach. I'll not go into the details, but we can definitely discuss that later during the workshop. But the idea is that as input, the model is receiving some frames of a video, and the model is supposed to be able to predict what happens next in the video. And specifically, it's using a contrastive loss function, meaning that the model is trained to make a contrast between the correct next frames and the wrong next frames. And everything is happening in the so-called latent space, so no reconstruction of pixel input is done here. So all the parameters in the model are trained end-to-end. And this convolutional part of the model is the part that we use after training. We use it as a model of the visual system, and we compare it with the visual system, with different parts of the visual system. So we started by just putting a 3D convolutional neural network as the backbone of the model here, and train it with visual prediction. And here we compared the neural network, our neural network, with responses from mouse visual system. So mouse visual system, similar to primates, is shown to have this kind of ventral versus dorsal type representations or pathways or specializations. The exact mapping is a bit controversial, is open to discussion, but we know that this sort of specialization exists. So we took this idea from the literature that some of these visual areas belong to the dorsal pathway, some others belong to the ventral pathway. We grouped these pathways and compared them with our neural network. So we took the most ventral areas of mouse visual system, and we compared them with our trained network. So here I'm showing the similarity of this area, this LM in mouse visual system, which is the most ventral type area in this specific data set coming from the open Allen Brain Observatory data set, open data set of mouse visual areas in response to different stimuli. So here I'm comparing this LM with different layers of my model, and we can see that the similarity goes up, saturates at some point, and either goes down or stays there, right? And the same is true for the other two areas here. Now let's compare them with the dorsal areas of mouse visual system. And here we see that the story is a bit different. Unlike the ventral type areas, the performance of the model on the dorsal areas is not very good. And we can see that as we go deeper in the network, the performance, the similarity is going lower and lower, meaning that whatever this network is trained to represent, however it's processing the input through the layers, it is getting farther and farther away from the representations of this area, the most dorsal type area in this data set of mouse visual system. So we thought that, okay, maybe one pathway architecture is not able to capture the two types of representations. So what we did was that we replaced the network with a network that has two segregated parallel pathways. The two pathways are identical and there's no difference between them. And we trained the whole system on visual prediction. And now we compared them with the two pathways of mouse visual system. And interestingly, we saw that, first of all, the two pathways learned different representations. They didn't learn the same representations of the visual input. They learned different representations. And interestingly, one of the pathways shown in blue was more similar to the ventral areas of mouse visual system, and the other pathway was more similar to the dorsal areas of mouse visual system. So this was suggesting that even with one single objective function, you can have a neural network that also mimics the specialization and automatically, spontaneously learns the specialization of the pathways in mouse visual system at least. So of course, this needs to be extended to other species, and also needs to be more closely investigated in terms of the limitations of the approach. So to summarize, it is important. So what I try to carry in this, to convey in this talk, is that specialization alignment between visual systems and neural network models of the visual system is quite important. We need to have a neural network that also has a specialization, and importantly, we also need to make sure that the model can also capture how these two pathways or these specialized networks are communicating with each other. How can they cooperate with each other to do a specific visual task? And this is another thing that needs to be done in future work. So we said that we can have two approaches. We can have per pathway losses, different losses per pathways. The ventral pathway was shown to be that's possible to model the ventral pathway object categorization. And also recent work shows that self-supervised learning alternatives of supervised object categorization can also give us a good model of the ventral pathway. And also self-motion estimation is also a good model of the dorsal pathway. But also we showed that our work shows that even one single loss might be able to explain both representations within the same network if we have this anatomical segregation between the two pathways. So of course, it's different studies use different species. Some relied on human data, monkey data, mouse data. We need to see if there is any species specificity in these results. Do we need to incorporate different architectural properties or different loss properties per animals? And this is something that, again, requires the studies that include multiple species within the same comparison. So that is it. And if you have questions or you want to discuss this more, please contact me. And I hope I'll see you in the workshop. Thank you.